Well, first and foremost, I'd like to thank Kayvon for the opportunity to address this distinguished group. Today, I'm going to tell you about the privilege that my group and I had in developing the first drug that slows down the progression of a human amyloid disease. I'll tell you a little bit about the strengths of this category of drugs, which now numbers 7, which is kind of amazing how fast this has developed, and the limitations in that one has to fashion a small molecule for each amyloidogenic protein. And in the later part of the talk, I'll describe the pharmacologic approaches we're using now to adapt the protein degradation systems that might be a more general strategy to address these diseases. Now, this whole story starts with the Welch Foundation when I was at Texas A&M, and that support really helped us get this project going. Thanks. So on this slide are listed the top 3 amyloid diseases or dementias in terms of numbers of patients affected. Of course, dementia is defined as the impaired ability to remember and think that's serious enough to impede the quality of daily life. Now, these diseases come in 2 categories. So-called sporadic, which simply means that the protein that all of us have, the wild-type protein, ends up aggregating and drives the pathology. Then, of course, there are inherited hereditary diseases where a mutation in one of the 2 alleles causes conformational instability and generates aggregates that drive these pathologies. Now, we became interested in the transthyretin amyloidoses not because they were the 3rd most prominent human amyloid disease, but instead because they are cell non-autonomous or they exhibit intrans proteotoxicity. What I mean by that is that the tissues that are ultimately degraded by protein aggregation don't actually make transthyretin. So transthyretin is only secreted from a few tissues. Firstly, it's secreted by the liver into the bloodstream, and aggregation of the transthyretin protein in the bloodstream to afford myriad of aggregates, including the cross-beta sheet amyloid fibrils, for which these diseases are named, destroys both the peripheral and autonomic nervous systems as well as postmitotic organs like the heart and the kidney. This protein is also secreted from the choroid plexus into the cerebral spinal fluid, which bathes the brain. And in the brain, the misfolding and aggregation of transthyretin destroys the parenchyma of the brain as well as the vasculature, which provides nutrients and allows waste clearance from the brain. Now, the protein is 25 times lower in concentration in the brain. And like Parkinson's disease, in the transthyretin amyloidoses, the periphery is affected first, about 10 years before the brain is affected. Transthyretin is also synthesized by the retinal pigment epithelial cells of the eye. And these patients have a number of eye problems, including glaucoma, and in unfortunate individuals, ultimately loss of vision. Now, the name of this protein gives away its normal function. It's a transporter of thyroid hormone, and only about 1% of circulating transthyretin does that because there are 2 high-affinity carrier proteins. It also carries retinol binding protein bound to vitamin A, and about 50% of the protein circulates as that protein-protein interaction. And it's the unliganded protein shown in the middle that puts us at risk for these amyloid diseases. Now, we thought there was enough evidence when we commenced this program to study aggregation as a means of conceiving a therapeutic strategy to intervene. So because this is a secreted protein, we first worked on the cell biology. And the bottom line is that for most of the hereditary sequences, they're secreted at wild-type-like levels with wild-type structures. It just so happens that they're conformationally unstable, and that puts the patient at higher risk for dissociation, misfolding, and aggregation that I'll have more to say about in just a second. Really unstable mutations, ironically, affect the brain first. And that's because the liver detects them as being problematic and degrades them instead of secreting them. So the brain tissue tropism for some mutations is a consequence of quality control by the liver, but not by the choroid plexus, which is kind of a bizarre result. Now, probably the most important and enabling discovery that we made was that the tetramer dissociation is rate limiting for subsequent aggregation. This is really slow. So this is a really stable protein. It takes on the order of a day in terms of its half-life of dissociation. But once it dissociates into these dimers, it can further dissociate to folded monomers. Now, most of the time, this equilibrium is occurring all of the time, and the monomer just snaps back to the tetramer because the driving force for that is immense. But either as a consequence of mutation or as a consequence of aging, for reasons we don't fully understand, sometimes the monomer can misfold. And this drives aggregation pretty efficiently into many structures, including amyloid fibrils. And there's now quite a bit of evidence that this ensemble of aggregates is the main drivers of these diseases. But how? So the numerous aggregates that are formed in the context of these amyloid diseases, including amyloid fibrils, compromise the membrane integrity of cells. And all of the components start getting mixed up. So cytosolic proteins start getting N-glycosylated. All kinds of bad things happen. And of course, the unique chemical environments of the mitochondria and the lysosome are also compromised due to membrane perforation. The innate immune system is also activated in these diseases. And initially, that protects us by clearing the aggregates. But ultimately, it attacks the synapses of the neurons and exacerbates the pathology. Of course, these aggregates consume the proteostasis network that's trying to keep the proteome in its right shape, location, and so on. And Ricardo's lab showed beautiful data that these aggregates sequester transcription factors, and that really screws up the transcriptome, which of course defines why a certain cell is a certain cell. So cells start to become less differentiated, and that's of course a huge biological problem. So it really appears like these neurodegenerative diseases or dementias are death of neurons by multiple cuts over decades of struggle. Now, if we delve a little bit more deeply into the rate-limiting step of transthyretin tetramer dissociation, what Ted Foss, a graduate student in the lab, showed is that of all the pathways by which this protein could come apart, it turns out it takes the bottom pathway. That is, the tetramer dissociates about this dimer-dimer interface. And this was a really good day in the lab because that's the unoccupied thyroid hormone binding site 1, and that's the unoccupied thyroid hormone binding site 2. So we rationalized that if we could fashion a small molecule that wasn't a thyroid agonist or antagonist that bound tightly to those two sites, we could stabilize the weakest interface in the protein and prevent the dissociation required for subsequent aggregation. And we argued that this was a conservative approach to go after these diseases because we didn't need to know exactly which proteotoxic structure was the main driver of pathology. And as a chemist, you wouldn't be surprised to realize that I didn't think it was the amyloid fibrils. That didn't make a lot of sense because they're buried deep in the interstitium between cells. I thought it was the diffusible soluble aggregates revealing all kinds of protein conformations and surfaces that could engage all of these pathways that I referred to you. But that was not at all what 99% of the thought leaders in this field believed. They all believed that it was the amyloid that drove these disorders. But fortunately, there were a few believers in the biotech sector, and they ultimately invested enough money to test this hypothesis in humans. Now, Tafamidis, whose structure is shown below Evan Powers, the postdoc who discovered its picture, is a small molecule that we conceive by structure-based principles. It binds really strongly to the first site, and binding to one site is sufficient for imposing kinetic stabilization on the entire protein. It's really fantastically bioavailable in that 61 milligrams once a day affords a plasma concentration of 30 micromolar. And if you forget to take the medicine for a given day, it doesn't really matter because the half-life's really long. So really an ideal small molecule that complements the structure of transthyretin beautifully. And while there are 150 crystal structures in the PDB of this protein, when we did the cryo-EM structure of this protein recently, we realized it was asymmetric, and we never saw that by crystallography. So beware when you're studying oligomeric proteins. Sometimes it makes sense to do cryo-EM, and I suspect that's why we had to make 1,000 small molecules to ultimately get at the answer. It should have been more efficient if we had exactly the right structure. Now, we call drugs like Tafamidis kinetic stabilizers. Why? Because they selectively bind the native state over the dissociative transition state, and they increase the activation barrier proportional to the binding constant. So I already told you that this protein dissociates with a half-life of about 18 hours. So you only need to add an additional kcal to the barrier, right? 1.5 kcals, a factor of 10, to make that half-life 160 hours, so basically to stop dissociation. So that's how these molecules work. They take a protein that can dissociate and turn it into one that can't by imposing 1.6 kcals in additional activation-free energy. And that locks the protein in its tetrameric form, but still allows turnover when it goes in the lysosome because acidic denaturation is so efficient. So while we were busy fashioning small molecules as kinetic stabilizers, including Tafamidis that turned out to be the winner, a very distinguished physician in Portugal by the name of Teresa Callejo was taking care of about 1,000 patients who had this VAL30 MET mutation. This is an inherited autosomal dominant mutation that causes peripheral and autonomic neuropathy. Age of onset around 30, pretty religious age of onset. Quite aggressive disease. And she figured out that some of her big families whose carriers she already knew weren't developing the disease. And she was smart enough to sequence them. And what she found is that those individuals who were protected had a V30M disease-associated mutation on allele 1 and a 3119 MET mutation in the other allele of transthyretin. And when we saw this abstract, we immediately thought, okay, this must work by thermodynamic stabilization, that is, the 3,119 met makes the tetramer more stable. That was true, but we completely got the mechanism wrong. So when Per Hammarström, who was a postdoc in the lab, started doing biophysics on these mixed tetramers, the fact that we could even make them tells you something important, that they're pretty stable. And so what he found is that proportional to the number of 3,119 met subunits incorporated into tetramers otherwise comprised of the V30M subunits, that you increase the dissociative transition state energy proportional to the number of these protective subunits. So you can increase the activation barrier by digging out the valley, right? That makes the hike harder. Or you can increase the mountaintop. And here we're increasing, well, Mother Nature, by virtue of this mutation, increased the mountaintop as a way of increasing the activation free energy. So you can either have a small molecule that binds to the native state to increase the activation barrier and lock the tetramer in a form that's not amyloidogenic, or you can increase the dissociative transition state barrier. And this was a really important result because we didn't have an animal model for this disease, and we were arguing to the FDA that we wanted to go into people and test this hypothesis. And this data turned out to make them confident enough to allow us to do a clinical trial. So the next challenge was to figure out what concentration of this compound tafamidis we needed in plasma to stop tetramer dissociation. So we made up this so-called subunit exchange assay to do this. The most important thing about this assay is that we can take tetramer from a person's blood, and in fact, we do the assay in the person's blood, and to it we add a recombinant transthyretin tetramer that just has a little acidic tag on it. So it turns out that in order for the tag subunits and the subunits in the patient's plasma to exchange, the tetramer has to dissociate. So that's rate limiting also for subunit exchange, just like it is for aggregation. And so what we'd love to see is, if we have enough drug on board, is that we block tetramer dissociation and we don't allow the subunits to scramble, so we don't see these peaks in the middle. The challenge is that there are 6,000 proteins in plasma, and this ion exchange chromatogram that I showed you, which works great in buffer, doesn't work so well in plasma because it's just a mess. So we came up with a small molecule that takes advantage of the fact that there's a PK perturbed lysine in the binding site for reasons we still don't understand, but it's really perturbed. It's down around 7. And we came up with a still bean that when it binds, it's not fluorescent, but after it undergoes a covalent reaction by thioester displacement, then you end up with an amide bond that freezes the still bean conformation. It renders the tetramer highly fluorescent. So now you can detect the tetramer in the sea of the 6,000 additional proteins in the plasma. And this is a Nobel laureate who developed wild-type sporadic TTR cardiomyopathy at age 85. He lived to be 96. And he was a very aggressive patient, and as soon as we had the safety data, we were doing dose titration in him. And we figured out that the 60 milligram dose of tafamidis was sufficient to stop subunit exchange. Now, in their infinite wisdom, and I say that sarcastically, the FDA forced us to use 20 milligrams in the first clinical trial. They were absolutely convinced, without any data to back up their hypothesis, that we were going to transform an amyloid disease into a lysosomal storage disease. That is, we'd make this protein so stable that it wouldn't be able to be turned over in the liver, which turns out to be nonsense, but nonetheless. But fortunately, even the 20 milligram dose slowed down progression, as shown by the bottom line, relative to the placebo group, which is shown on the top line. Now, since this was the first clinical trial ever done for a human amyloid disease, we had to guess what endpoint was going to give us a statistically significant answer. And we took advantage of Peter Dick's insights from the Mayo Clinic, wherein he hypothesized a neurologic impairment score that was used for diabetic neuropathy. The problem with that is every single trial for diabetic neuropathy failed. But the neuropathy in this disease progressed a little faster, and the physicians thought that it would work, and of course they guessed correctly. So this was approved by the EMA right away, and again, the brilliance of the FDA comes back and said, well, you know, even though we told you guys that doing a trial in V30M patients was really forward-thinking, now we disagree because there are other mutations in the US, so go do another trial. Well, fortunately, Pfizer was interested in buying FoldRx, the company who helped us develop this, and they did that. Why? Because they were interested in the sporadic disease. There are several million patients who have the sporadic disease, and they did a really nice trial, which I'll tell you about in a minute. But we learned something very important from the hereditary polyneuropathy trial, and that is, if you started out on Tafamidis, about 70% of those in the study had very little or no disease progression. But if you were simply on placebo in a demographically equivalent group for 18 months before you switched on to Tafamidis, now only less than 50% of the population responded. This has turned out to be true in every single amyloid disease clinical trial since. Starting early is really critical in these patients. And so, as you might imagine, there's a lot of effort now on early diagnosis. And since Tafamidis in 4 years will become placebo, a lot of people are now, especially for hereditary polyneuropathy, thinking about prophylaxis, which I think is the ultimate way to treat these disorders. And while I don't have time to talk about it today, we repurposed a Merck NSAID, which is in more than 50,000 patients for being used for prophylaxis now. It's not nearly as good as Tafamidis, but it's good enough to prevent disease onset. So, Pfizer. Pfizer, along with Matt Maurer, came up with a really nice cardiomyopathy trial. It was really first in many ways. They used a very clever statistical method called the win ratio, which is now used in many clinical trials. And they decided to look at 20 versus 80 milligrams. And the endpoint was a reduction in all-cause death and the frequency of cardiovascular hospitalization. And as you can see from the p-value, the trial worked really well. And there was, again, a huge advantage of being on placebo or being on drug right from the beginning versus being in the placebo Tafamidis group. And it's really amazing now that these Kaplan-Meier curves, which of course reflect patient deaths, have flattened out dramatically. And that's simply because these patients are no longer in stage 2 New York Heart class anymore. They're being diagnosed earlier. For those that are being diagnosed, it's still a difficult disease. So your risk of reduction of death is 65% if you're class 1. If you're a little later stage, it goes down to 40. So again, early detection and early treatment is absolutely key. There are currently 60,000 wild-type cardiomyopathy patients on Tafamidis, and it's clearly a drug that extends lifespan and health span. Now let's get back to the mechanism. So today we have methods for detecting a lot of these non-native structures, including amyloid fibrils. So we can start to address what are the main drivers of pathology. And it turns out that subsequent to the introduction of Tafamidis, the kinetic stabilizer, there are also drugs that degrade the messenger RNA of TTR and drop the protein concentration so low that it can't aggregate anymore. Those are called the silencers, and Alnylam and Ionis have introduced those. So all of these drugs do the same thing. They take out all the aggregate structures except the amyloid fibrils. The amyloid fibril load does not change in these patients for several years, and probably never, or at least on any kind of relevant time scale. So I think what this pharmacologic data tells us is that these diffusible non-native structures are the main drivers of pathology. Now, I don't want to leave you with the notion that the amyloid fibrils aren't pathologic. They clearly are. They stiffen the heart, and they replace normal tissue by collagen or fibrotic tissue. And in cases, really rare cases, about 1 in 1,000, where a patient's immune system ends up clearing the fibrils by recruiting macrophages through producing the right antibodies, in those cases, the patients do miraculously well. It's almost like you clear the amyloid and you turn off the disease. The problem is we don't know how to do that with monoclonal antibodies yet. And most of the trials for clearing amyloid fibrils aren't working because we're replacing the amyloid fibrils with collagen, or we're eliminating the extracellular matrix, both of which aren't so ideal. But eventually, that will get figured out. Now, I mentioned to you that initially we didn't really have a problem with the dementia in these patients because they didn't live long enough. But now they're living much longer. And as a consequence of having biologics that don't get into the brain, fortunately Tafamidis does, we're starting to see in patients treated with biologics after about 10 years quite a bit of dementia and amyloid in the brain, as you can see by these PET images. These are just small molecules that intercalate into the amyloid fibril and reveal a signal by PET. Now, the main problem is that these patients have focal neurologic episodes. That means they lose consciousness from a few seconds to several minutes. Clearly, that completely changes your quality of life. And so, hopefully, Tafamidis or its successors will do an even better job of tamping down this phenotype in the future. And these patients also have stark dementia as a function of age, as a consequence of aggregation in the brain. Now, it turns out that you can use the subunit exchange assay, to show that while Tafamidis at the 20 milligram dose only occupies about half of the transthyretin protein in the blood, hopefully at the higher dose, which is used in all patients now, that the stoichiometry is closer to 1:1. We still haven't talked the patient into doing a lumbar puncture to get this data. I hope we can ultimately do that. So I want to close this portion, and the next one will be quite brief, just saying that we now have at least 7 medicines for treating transthyretin amyloidosis. Tafamidis was approved 7 years before the others. All of these drugs work pretty well. So the argument isn't which should I be on. You should be on one of them. And the whole game now is to identify patients early, and a lot of efforts going into that. And of course, if we had a depleter that could take advantage of the immune system like those rare patients, that would be incredible, because then we could reverse the disease. We're not there yet, as I mentioned. Now, even 5 years ago, there were papers in Science, Nature, Cell, and pretty much every medical journal that aggregation doesn't cause the degeneration in amyloid diseases. People got really discouraged because of all the failures in Alzheimer's disease clinical trials. I think clearly now that's not true. We even have 2 antibodies now that clear the amyloid fibrils in Alzheimer's patients and slow down disease progression. Those drugs are not awesome, but they're pretty darn good, and I think with time, we'll learn how to use them more effectively. So we've been working hard on making kinetic stabilizers for what is probably the 5th most prominent amyloidosis, which is called light chain amyloidosis. Unfortunately, this is both a cancer, so the plasma cells start to proliferate, and instead of secreting light chain and heavy chain to make antibodies, they only secrete the light chain. The light chains in those patients are really unstable, and they go on to make a plethora of aggregates. So the way these diseases are treated today is using chemotherapy agents. And that works okay for about 40% of the patients. We decided that we would, well, this isn't supposed to be bound here, it's supposed to be bound here. But the bottom line is we decided to make a stabilizer, and with a lot of effort, we made one, and we're almost through phase 1 clinical trials now. And so hopefully we'll be able to test this in the clinic in a couple of years. But what's the problem? It takes a lot. This particular light chain stabilizer, we had to make 3,000 small molecules before we ended up with a drug candidate. And you have to make a specific small molecule for each of the 40 amyloidogenic proteins. It's an arduously slow process. So what are we doing now? We hypothesized that the autophagy lysosome pathway could be a nice way to find a single small molecule to treat all of these diseases. That is, instead of treating the supply side of the aggregation equation, we're going to start working on the clearance side, because we already know that there are receptors in our body that recognize non-native proteins and get rid of them. And the pathway that mainly does that is called autophagy. So you end up with a double membrane structure studded with receptors that bind to non-native proteins, and this closes into a double membrane structure called an autophagosome that merges with the lysosome that comprises hydrolytic enzymes that can break all of the macromolecules down into their monomers. The acidic environment of the autolysosome is incredibly efficient at getting aggregates back to monomeric structures so the enzymes can degrade them. So I think this pathway, if we could learn how to tap it in the elderly, will be a fantastic approach to have one small molecule that could work for most of the human amyloid diseases or dementias. The human genetics screams that this should work. So there are mutations in virtually every step of the autophagy lysosome pathway that modestly diminish efficiency and upon aging lead to a wide variety of neurodegenerative diseases, including some of those that I showed you. The problem is that this is a cellular pathway, and it's really hard to do high-throughput screening to come up with chemical leads. But there was a really nice paper that came out of New York and Harvard suggesting that the autophagy lysosome pathway is the main pathway for clearing lipid droplets. And this paper caught our attention because loading up cells with lipid droplets is really easy. You just add fatty acids to the media. They convert the fatty acid to triglycerides. There are fantastic small molecules like Bodipi that light up the lipid droplets, and so now we can do a high-throughput screen, and we did this at Peter Schultz's Caliper high-throughput screening facility, and we came up with a couple hundred really quite useful hits that got rid of the lipid droplets relative to the placebo control. Now, the assays in autophagy are a nightmare, and that's because there isn't one rate-limiting step. There are a whole bunch of different biological processes that ultimately lead to lysosomal degradation of these aggregates. And so you don't know at what step your small molecule is working. But one of the assays that's quite useful is to monitor this protein called LC3, which is the protein that tethers the membrane, the phagophore membrane, to the receptors. So ultimately, LC3 is degraded in the lysosome like all the other proteins that are there, and so you can tag it and watch its degradation. And a really nice way to do that is photoconversion. So you fuse LC3 to a green fluorescent protein, you then flash it with a laser to make it red, and then you watch how fast the red protein disappears. Really convenient way to do a pulse chase experiment, and you can show that you can decrease the half-life of LC3 as a function of the concentration of one of the hits here, which we call A5. You can also look at this using a ratiometric assay, taking advantage of the fact that GFP is pretty sensitive to acidification where RFP isn't. So over time, if these bands become broad and red, that means that you're activating autophagy, and if the bands are broad, you have a nice dose-response curve as well. I don't believe in anything in autophagy until I can see an electron micrograph that shows that you're making double membrane structures that are electron dense, and you can see that the hits that come out of this assay really turn on this pathway. Now, most of the drugs that are out there today that turn on this pathway are mTOR inhibitors. And the problem with mTOR inhibitors that are starting to emerge is that they induce type 2 diabetes in a good fraction of the patients being treated with them. And of course, they're also immunosuppressants. So we've put a lot of effort into getting away from mTOR inhibitors. Not that mTOR inhibitors can't possibly work, they can, but we just decided that we would take a different path. So basically today we use phosphoproteomics to look at all the downstream targets of mTOR to show that we're not inhibiting it, that is, that the downstream targets get phosphorylated. Now we know how the end of this pathway works. This molecule A5 sends a transcription factor by the name of TFEB into the nucleus, which reprograms the lysosomal pathway to be more active. So you can see pretty clearly TFEB is in the cytosol, you treat with small molecule, and now it's in the nucleus. Now I want to just end with some pathobiology that's changed by this small molecule, that is this autophagy activator. So the problem in many of these neurodegenerative diseases is that the aggregates end up in the axons. And the aggregates are so big that they prevent the trafficking of critical cargoes in both directions that ultimately have to get to the synapse in order for the neuron to work. So with one of my colleagues, Sandra Ancolata, we came up with a nice assay where we transfected in a neurodegenerative disease protein. You can see just by virtue of this movie how quickly the cargoes are moving both toward and away from the cell body on the left, and how slowly they're moving on the right, if at all. So we want to correct that. And fortunately, this small molecule A5, above a dose of about 50 nanomolar in a dose-dependent fashion, quite rapidly clears these aggregates out of the axons by sending them back to the cell body for autophagy lysosomal degradation. And you can see this quite nicely in the chymograph. So these chymographs are just movies of the vesicles moving along, and so you want to see these streaks that go in either direction. The disease state is this, that is, the aggregates are present and there's not much moving, and after drug treatment, you largely restore vesicular trafficking. This is a huge advance because this is one of the major problems that are central to all neurodegenerative diseases. And fortunately, recently we've been able to identify the target of this small molecule. It wasn't a target that we expected. In fact, our collaborators at UT Southwestern said there's no way this is the target. But they're really good at genetics. In a couple weeks, they called me up and they said, I think you found the target. Because when they knock down this protein, it really activates autophagy. So it's a good negative regulator. It's in the Golgi. It's a new pathway, and we ultimately have to figure it out. All right, so I think it's clear now that, in my mind, neurodegenerative diseases are not going to overtake cancer as the second leading cause of death. And that's because we're moving fast on the pharmacology now. And there are going to be drugs soon for even better drugs for Alzheimer's, and hopefully we can get there with Parkinson's soon. I haven't run a pipette in a long time, and so these are the people who are critically responsible for Tafamidis and the assays that are involved. And the highlighted people on this slide are working on the autophagy lysosome project in the lab right now. And mostly I'd like to thank you for your kind attention, and I'm happy to address any questions that you might have. Thank you.
Lead it off with a question. Excellent talk. I'm curious what the obstacle you think would be to applying kinetic stabilization to amyloid beta or alpha-synuclein or other amyloid diseases. Is this because you have this nice steroid binding pocket with transthyretin? And is there ways that we could use other principles to apply that elsewhere?
So there are two types of amyloidogenic proteins. There are those that have a well-defined structure, like light chain and transthyretin, alpha-synuclein, at least when it's bound at the membrane. And then there are proteins that are intrinsically disordered, like A beta. It's not easy to directly target A beta, whose aggregation is linked to Alzheimer's disease, but we can target protein-protein interactions to stabilize them with small molecules, and several companies are now doing that, both for Alzheimer's disease and for Parkinson's, as a way to kinetically stabilize intrinsically disordered proteins.
Over here, to your left.
Thank you.
It's a bright light. Super beautiful work. I was struck by your comment on the death of some of the nervous system by many cuts, and one of those is the innate immune system. And in particular, you described it as sort of a duality, right? At first it's protective, and then eventually it's doing harm. Do we have an understanding of, are there qualitative differences between when the innate immunity is acting in that first way versus second way, or is it a thresholding problem? Speaking to that, maybe it will help some of the understanding the longitudinal effects.
Yeah, so that's a great question. So the way that this works in the simplest minded way is that you have an antibody that recognizes the non-native structure. That antibody, through its effector domain, recruits microglia and macrophages to the insult to get rid of it. And you're right, initially that's protective. So you can look at the transcriptional program in the macrophages and microglia when the immune system is being helpful and it was starting to take out synapses, which you don't want. The transcriptional programs are clearly different. What we don't yet know is exactly how to control those transcriptional programs, but that understanding will come. Denali, for example, is working on that quite aggressively in Alzheimer's disease by modulating TFEB. Modulating TREM.
Thank you.
Hi. It was interesting hearing that cryo-EM and crystallography missed some things in your protein structure. I was wondering if you were able to use any mass spec for looking at dissociation, whether with your drug development or looking at tetramer subunit structures, either just mass spec and ion dissociation or like mass spec imaging.
I perhaps went too fast through the cryo-EM thing. So I don't think there's any question about the crystallography being interpreted correctly. It was. All those structures are symmetrical, whether there's no ligand bound, one or two. But that always bothered me because the binding is clearly negatively cooperative. So the structural biology and the biophysics didn't match, right? And then a couple years ago, Gabe Lander, who is pushing the limits for how small a protein you can solve structurally by cryo-EM, wanted to do TTR, transthyretin, because it's right at the limit. And he called me up and said, there's a little problem here. Every single structure we have is asymmetric. I'm like, that's awesome. That aligns with all the biophysical data. So There are a number of examples of this now, where when you do crystallography on an oligomeric protein, you have higher symmetry than actually what exists in solution, presumably as a consequence of the crystallization process. So the take home is, if you're working with an oligomeric protein and you're doing structure-based drug design, you should really try to do cryo-EM if at all possible. Of course, mass spec is incredibly useful, so.
Thank you.
Hey, Jeff. Well, there was Nadia Small. Actually, she is a first-year graduate student at Texas A&M. I just want to let you know. I have a question related to the small molecule itself. Of course, your small molecule is really targeting a protein which can activate the whole autophagy pathway, right? So, but there are people also developing the Lita type of technique. How do you compare the two, the two ways to degrade the protein?
If I understood your question correctly, you can use a Stewart-Schreiber type approach where you can recruit things to the lysosome through making bivalent small molecules, or you can take advantage of the inherent nature of the evolved receptors to recognize non-native proteins.
Right.
And since there's so many good groups already working on harnessing the autophagy lysosome pathway with small molecules, we decided to take advantage of the receptors as another means, a complementary means to do that. We're not going to know which is going to work better until we get further down the road.
Good, great, thanks.
But what is clear is that autophagy activation doesn't affect the proteome if you do it right. That is, you don't want to trigger this pathway every single day. We're talking about treating like once a week, and that seems to be the sweet spot. And when you do it that way, you only get rid of non-native structures, not regular proteins that aren't supposed to be degraded. Thank you very much.
One more quick question, Jeff.
Oh, sorry, where are you?
Right here.
Okay, sorry, Kayvon.
The fact that it's asymmetric and Tafamidis is negatively cooperative now that you've got the structure and the biophysics lined up, does that mean there's a potential for a two-drug cocktail to do a better job of trapping? Sort of maybe like we'll hear about from Ju and CFTR?
Yeah, I think that could be quite interesting. I mean, we're pretty confident that occupancy of one binding site is sufficient to make the barrier high enough that you don't see dissociation. Okay. Now, that's not to say that you couldn't make a small molecule that could start to use 1.5 or 2 sites. I think that could be possible. And what you're suggesting is really interesting. That is, could you have 2 different molecules that were tuned in terms of their cooperativity to load up both sites? That's a really clever idea. That's not being pursued right now, but I think that would work.
Okay.
Thank you.